The sooner we get going, the sooner we get done, right? Big weekend plans? Studying biochemistry. So I am planning a review session like I did last time after class on Monday. So I'll do a review session after we finish class on Monday. Okay? All right. And I will videotape that as before. Okay, we've got quite a bit of ground to cover, so I'm going to just dive right in. Last time I talked about energy considerations, and it's important that you understand basic calculations of energy. So if you're having questions or concerns about those, please come see me. I will tell you that it's important to understand the distinction between what delta G is and what delta G0 prime is. That's the most common confusion that students have, the difference between delta G and delta G0 prime and what those mean. Okay? So if you have questions about those or anything, of course, as always, please do come and see me. The things I talked about last time were introducing the concepts of catabolism and anabolism. And I'll remind you that catabolism is a process involving breaking down larger molecules to smaller molecules. It usually involves oxidation, and it usually generates energy. So we break down proteins to amino acids, we break down carbohydrates to sugars, we break down fats to fatty acids. Okay? In the case of anabolism, we're building things up from small pieces, we're making things, and to make bigger things from smaller things requires input of energy. It also involves usually reduction as well. Okay? That energy comes from the foods that we eat. That's why we have to eat food, so we can make things that we need, like proteins and so forth, in our body. All right. This figure very briefly shows different states of oxidation of carbon. And it's these different states of oxidation of carbon that are largely the sources of the energy that we have in our body. On the left, you see the most reduced form of carbon, and on the right, you see the most oxidized form of carbon, which actually is missing a C and O's. I just noticed that. Okay. There should be a C in the middle and two O's, meaning carbon dioxide. All right. As you oxidize carbon, energy is released. And, of course, as we're going through respiration, one of the byproducts of respiration inside of us is carbon dioxide. So we're releasing carbon dioxide as a result of the oxidative processes that are going on inside of our body. This figure shows the relationship between oxidation and reduction. And if we look at this, we see on the left we have an alcohol, in this case ethanol. And on the right we have an oxidation product of ethanol. In this case, it's acetaldehyde. And of course, you know from organic chemistry that going from an alcohol to an aldehyde involves involves oxidation. And oxidation involves loss of electrons. So you're losing electrons in this case, and so you see the 2 electrons here and 2 protons that come off as well. As I've mentioned in class earlier, electrons are reactive. Electrons aren't things you want floating around uncontrolled, and so that's for that reason that we have the electron carriers that I talked about in class. And those electron carriers are very important, not just from a protective standpoint, that is, protecting them from causing reactions and going on inside the cell, but also in terms of conserving the energy that's released in that oxidation. So we'll see that those electron carriers carry not only the electrons, but the energy associated with that oxidation to the mitochondrion, where that energy can be converted into ATP. Okay. Well, here's one of those carriers, or actually 2 of these carriers, in the form of NAD, which is shown on the left. NADP, of course, is if you put a phosphate on at that point, and no, you don't need to know the structures of these molecules. But I'm just showing you that what's happening here is we have a reduction that's going on from left to right. And what's being added is a pair of electrons and 1 proton. All right? A pair of electrons and 1 proton. So what you see on the left is what we call NAD+, or if we have a phosphate, NADP+. And what we have on the right is NADH, and there's no plus. And the reason there's no plus is we've added 2 negative charges and 1 positive charge to something that had a positive charge. So if we have add a minus to a plus, we get a 0, and that's why the charge of NADH is 0. Okay. FAD is a molecule that's a little bit more complex than what's shown here. That's why you see the R coming off here. But the nuts and bolts part of FAD is shown on the screen. This is where the electrons are accepted and protons are accepted. And you can see the conversion from going from this structure here on the left to this structure on the right. You can see the addition of electrons to convert that double bond into a single bond. And you see the addition of 2 protons. In this case, we start with something FAD that has no charge, and we're adding 2 electrons and 2 protons. That's a net gain of charge of 0, so we start with 0 and we end with 0. On the right, we have what's called FADH2. In order for this reaction to occur, or the other reaction that I showed you on the last slide, the conversion of NAD to NADH, in order for either of those reactions to occur, there has to be a source of electrons. And the source of electrons is an oxidation reaction. So as I've noted before, for something to be oxidized, something else will be reduced. In this case, the reduction is this FAD. It could be the NAD getting reduced to NADH. It doesn't really matter. But to have something reduced, there has to be a source of electrons. Okay. And there's the full structure of FAD. And the next thing we want to consider with respect to energy and metabolism is this guy right here. We've talked about it in class, and you hear about it ever since your beginning biology class, and that's ATP. ATP is what I refer to as the gasoline of the cell, and I think I've actually said what I'm getting ready to tell you here previously, so again, I'm going to say it quickly. And that is that, of course, ATP is a high-energy molecule, and the reason it's a high-energy molecule is because these phosphates that are sticking out over here all repel each other. However, they're covalently bound to each other as well. So that means that there's like a spring that someone holds together. There's potential energy. If you release the energy of the phosphates, that energy will be available to the cell to do things. Just like if we release that spring that we've got coiled, it will spring out on its own and release energy. And so that's what's really happening when ATP gets converted into ADP. The spring is being released, and it's letting go the potential chemical energy that it has stored within itself. Okay, ATP, ADP, AMP. All right. ATP, of course, is the sort of conversion form of energy. It's what we refer to as energy currency. So currency is like money, of course, and money is accepted in virtually any place that you go. If you want to purchase something, you need money. And money works as a currency because it's universally accepted. Okay? ATP is compared to a currency because it's universally accepted within inside of a cell. ATP generated in one set of reactions is used in other sets of reactions, and it's used pretty much universally. We've only seen one exception to the energy of ATP so far, and that was the use of GTP in the process of translation. So that means we need to have sources of ATP, and so what you see on the screen is how that ATP is generated. Here are molecules that are large molecules that are broken into smaller molecules. This is catabolism going on from left to right, starting on the top. And the energy of that release in the form of catabolism is captured by the cell in the form of chemical potential energy in the form of ATP. ATP then can be broken back down into ADP, and that energy of that release is then used to drive the synthesis of cellular macromolecules like proteins, like DNA, like RNA, and like carbohydrates and fats. And that takes energy to do that. So we have to eat up here so that we have nutrients to produce the ATP to make the things that we want down here. Okay. One of the things that we'll talk about with respect to metabolism is the need to recycle electron carriers. This is a very important concept, the need to recycle electron carriers. Okay? Cells have limited amounts of electron carriers. So when we go from NAD to NADH, we can't just forget about it, because the more NADH we have, the less NAD we have. And if NAD is needed for reactions that are oxidative in nature, and it is because NAD is accepting those electrons, if NAD is needed for those reactions and there's very little NAD available in the cell, what's going to happen to those reactions? Those reactions aren't going to go. And if those reactions don't go, we have no source of energy, and the cell is in deep doo-doo. So cells have ways of recycling that NAD to NADH, and we'll talk about a couple of them. And the way that they recycle them depends upon whether or not the cell has oxygen. It's a very, very important central concept to understanding the phenomenon of metabolism, and we'll talk about that. That's actually occurring in the electron transport system for the most part. Okay. Well, all that buildup is to get you to the point where we can talk about the first of our metabolic processes. And the first of those is glycolysis. Glycolysis literally means breakdown of sugar. Glyco meaning sugar, lysis meaning breakdown. So glycolysis is the process whereby glucose is broken down into simpler molecules. As you will see, there are 10 reactions that are involved in glycolysis. And those 10 reactions release some energy, but not most of the energy of the sugar. That's interesting. It releases some of the energy, but not most of the energy. Most of the energy of the sugar is actually released later in the citric acid cycle. So some of the energy is realized in the breakdown of glucose in glycolysis, but not most of it. Most of it comes from the process of the citric acid cycle. Well, one of the products, actually, let me come back to that. I want to start with the big picture. The big picture of glycolysis is what you see on the screen. Now, the first question students have is, do I have to memorize every step? Do I have to memorize all the structures, et cetera, et cetera? Okay. I think that you should know the names of the intermediates. You should know glucose goes to glucose 6-phosphate. You should know glucose 6-phosphate goes to fructose 6-phosphate. Okay? Now, there's 10 of those, right? Yeah. And you're welcome to know any abbreviation that I use. Yes, you're welcome to use it. You can call it G6P. Yep. G6P is fine. Yep. So you're welcome to use any, and that's true for anything in the class. Anytime I use an abbreviation, you're always welcome to use the abbreviations I use. Okay? Okay. Now, I'm just going to sort of walk you through this in words in terms of what's happening. Okay? You get some of this I think, in basic biology, but we'll cover it in a little bit more depth than you'll get in basic biology. Okay? If we look at this process, there's really, I like to think about it occurring in 2 phases. Some books will break it down into 3 phases, and all these are just semantic arguments. There's no absolute about any of these things, all right? But really 2 phases. One phase is what we call the energy investment phase, and it's in this phase that the cell actually has to burn energy to start the process. In other words, it has to burn ATP to start glycolysis. The second phase is the energy realization phase. And it's at this point that the energy is realized. You get more energy out in the second phase than you burn in the first phase. So there's a net gain of energy in the process of glycolysis. Now you see that this has a Y shape, And it has a Y shape because you start with a 6-carbon molecule glucose, and you break the glucose down into 2 molecules of 3 carbons each. Fortunately, the 2 molecules you get are identical, and essentially everything here is identical. There's one step that's not identical, and then everything else is the same. So if you learn this, you know this. Okay? So you're not going to have to learn 20 structure. You're not going to learn structures anyway. The only structures on here you need to know are those that you already had to learn, which were glucose, right? So if you know glucose, the structure of glucose, and I said draw the structure of glucose 6-phosphate, you should know how to do that because you know the numbering on the glucose, and you say, oh, we put a phosphate on number 6, I've got it, right? Fructose 6-phosphate. If you know the structure of fructose, you can put a phosphate on position 6, and you would know the structure of fructose 6-phosphate. But you don't have to know the structure of 2-phosphoglycerate or any of those. I'm not making you memorize those structures, okay? Okay. So, down here in the lower part, this is where we realize the energy for glycolysis, okay? The lower part includes one step, repeated in each one, but one step of oxidation. There's only one oxidation step in the entire glycolysis pathway. Okay? Only one oxidation step. And how do I know it's an oxidation step? Well, I told you earlier in the class, and I'll remind you here, anytime you see NAD being converted into NADH, that is an oxidation because that's a source of electrons. And those electrons are coming from the molecule in glycolysis that we'll talk about. Okay, at the very end, we get 2 pyruvates. At the very end, we have had a net gain of 2 ATPs. We've also had, at the very end, a net gain of 2 NADHs. And these turn out to be pretty important later. So we don't want to throw those away. We don't want to forget about those. And it's this step right here where you see the NAD being converted into the NADH that's critical for the cell to have enough NAD. When the cell starts running out of NAD, all you fermenters should know this, what happens is it finds an alternate way to make NAD, and that alternate way to make NAD is how we get ethanol. That's what the fermentation in yeast is all about. Now I'm going to tell you something that may surprise you, and that is fermentation is going on inside of you. It's going on inside of you every time you exercise. But it's not what you think it is. Okay? We'll talk about that in a bit. All right. Well, that said, now that I've got you thinking about getting drunk while you're running, which you won't be, okay? Let's talk about the process. The starting material for glycolysis is of course, glucose. And glucose, I've told you, is a poison. Cells treat it like it's a poison. What do I mean when I say it's a poison? Well, let's think about this. A person who has diabetes is suffering ill effects because glucose is a poison. And it's suffering ill effects because their blood glucose levels are rising because their insulin is not managing that level appropriately. The cells are making insulin, and the message to cells when they make insulin is take glucose into yourself. When cells take glucose into themselves, they're reducing the blood glucose levels, but they've just taken in a poison, and they need to do something with that poison. As I mentioned earlier, they will either break that down in the process of glycolysis, or they will convert that glucose into a storage form like glycogen or starch or something like that. Our cells, of course, don't make starch. They make glycogen. All right. So this is showing how one way that cells are reducing their glucose concentration. And cells don't have much free glucose in them, for the reasons I've stated. First reaction, catalyzed by an enzyme named hexokinase. Any enzymes I name are fair game. So you should know that hexokinase converts glucose into glucose 6-phosphate. converts glucose into glucose 6-phosphate. This is one of the steps that requires input of energy. Because it turns out, as I mentioned the other day, that putting a phosphate on glucose creates a molecule that has a higher energy than glucose does. Glucose 6-phosphate has a higher energy than glucose. Well, how do I make a molecule that has a higher energy than glucose? It means I'm going to have to put some energy in, and the energy comes in the form of breaking down ATP to ADP, and the phosphate gets put onto position 6. The second step of glycolysis is a very simple step, and guess what? I'm not going to tell you the name of the enzyme. You can read it there, and if I don't tell you the name, then I'm not holding you responsible for it. How's that? Everybody happy with that? The second reaction of glycolysis, fructose 6-phosphate is getting glucose 6-phosphate is getting converted into fructose 6-phosphate. Glucose 6-phosphate to fructose 6-phosphate. This is what we refer to as an isomerization. We're just simply rearranging the molecule. We're not doing anything but rearranging the molecule. The next step of the reaction, is another step that requires input of energy because you're making a molecule that has a pretty fair amount of energy in it. The molecule that you're making is fructose 1,6-bisphosphate, and unfortunately I have to tell you the name of the enzyme here. It's phosphofructokinase. But you can call it PFK if you want. PFK. The reason I tell you the name of that enzyme is because that enzyme, we'll see later, is very important for controlling whether or not glycolysis moves. That is, whether it goes in the glycolysis direction or not. What does it mean to say the glycolysis direction? Well, we're going from top to bottom, from glucose down to fructose. From glucose down to pyruvate. That's the glycolysis direction. If we were to start at pyruvate and go upwards and make glucose, we would be going in what's called the gluconeogenesis direction. We'll talk about that later. But right now we're going in the glycolysis direction, which is going from glucose, a catabolic pathway, down to pyruvate. Yes? So you're making a molecule called fructose 1,6-bisphosphate. It's right there. And you can call it, by the way, I don't like their abbreviation here. I tell you what, you can call it that abbreviation. How's that? That'll keep it simple. FBP. Okay? I usually like to call it F16BP, but you can call it FBP. Because it's Friday, right? All right. Well, at this point, we started with a 6-carbon molecule. We still have a 6-carbon molecule. In the next step, we're getting ready to go in that Y. We're going to split the pathway. And not only are we splitting the pathway, but we're splitting the molecule in half. We're going to break that fructose 1,6-bisphosphate into 2 molecules. Each of those molecules is going to have a phosphate. If you look at this structure right here, you can see there's a phosphate on the top, and there's a phosphate on the bottom. And so if we break this guy in the middle, we're going to have 2 molecules of 3 carbons, one of them here and one of them over here. Okay? Here's another enzyme I got to tell you. Aldolase. A-L-D-O-L-A-S-E. I tell you this enzyme because of all the reactions I've just told you, this reaction is energetically unfavorable unless the cell does some tricks. It's energetically unfavorable unless the cell does some tricks. What do I mean when I say that? All right? It means that if I start out with equal concentrations of the reactant and the products, what's going to happen is that the reaction is not going to go forwards. It will have a positive delta G 0 prime. And therefore, the reaction has a positive delta G 0 prime, and therefore it has a positive delta G, because if I have equal concentrations of everything, the log term is 0. The delta G 0 prime is positive, which means that the delta G will be positive, and that means reaction isn't going to go forwards, it's going to go backwards. So this reaction is the most interesting reaction of glycolysis. It's the most interesting because here's a pathway that's going to generate a lot of energy, but here's this big hill in the middle of it that the enzymes have to get over before they can release energy later. Very, very important that they get past this enzymatic reaction. The products of this reaction are 2 molecules of 3 carbon each. 1 dihydroxyacetone phosphate that you can call DHAP. Second, glyceraldehyde 3-phosphate that you can call G3P. Okay? Now, at this point, the cell does you a favor. The favor it does you is that the DHAP gets converted into G3P. So everything at this point is just 2 molecules of G3P going downwards. Make sense? We had 1 DHAP, we had 1 G3P, but the cell converts the DHAP into G3P, so now we have 2 molecules of G3P, and now everything's going to be 2 copies of everything all the way down. So we've got to double everything. That's why we're going to have 2 pyruvates at the end. Okay. Let's see. What do I want to say here? There's the actual reaction. Up close and personal. Okay. All right. Well, we're now ready for the energy phase of glycolysis. The energy realization phase, I think is what I called it. At this point, we have 2 molecules of glyceraldehyde 3-phosphate. Okay? The first reaction is the only oxidation reaction that occurs in glycolysis. Okay? The first reaction is the only oxidation reaction that occurs in glycolysis. And I know I say I'm not going to tell you all these enzyme names, but I have to tell you this one too. It's got a long name. You can call it G3PDH. There's only one more enzyme name you'll have to know after this one, okay? If that makes you feel any better. G3PDH. G3PDH converts something that's an aldehyde that you can see right here, that is glyceraldehyde 3-phosphate is an aldehyde. It's converting it into an acid, and it's making an ester because it's linking it to a phosphate. Now, I'll tell you a secret that I ask as a question in my 450 class, and my students get wrong all the time, and you'll know the answer before I ever ask it. Okay? The question I asked the students in my 450 class is, I said, well, let's think about this. We went from glucose to glucose 6-phosphate. What did I say about putting that phosphate on? It took energy, right? And when I went from fructose 6-phosphate to fructose 1,6-bisphosphate, I said it took ATP energy again, right? And now I'm showing you a reaction where we're going from glyceraldehyde 3-phosphate to a molecule that has 2 phosphates, and there's no ATP. How's the cell doing that? Well, most people kind of sit there and they scrunch at it, and then there's usually 1 or 2 people in the class who will answer the question, and I'll tell you the answer to the question. The answer to the question is, where does that energy come from? The answer is, it comes from that oxidation. Oxidation releases energy. And I go, oh, yeah, that's right. Oxidation releases energy. So that's why we see oxidation and the addition of a phosphate in the same step. The cell is putting a phosphate on without having to waste an ATP to do so. Yes? Electrons are being lost. You can see them being lost right there. right? Exactly. And those electrons being lost mean it's an oxidation, and oxidation releases energy. Yeah. All right. At this point, we have a molecule that has a lot of energy. A lot of energy. Okay? If I have something that has a lot of energy, I can do something with it, right? If I have a can of gasoline and I put it into my automobile, I can make my automobile move and take me places, right? Everybody knows that? That high energy is useful for some purpose, right? The high energy in this 1,3-bis phosphoglycerate, and by the way, you can call this 1,3-BPG. 1,3. Does that sound like something you've heard before? Okay. We're getting close to that, actually. Okay. 1,3-BPG has a lot of energy. The energy of that 1,3-BPG can be used to make ATP. And that's what's happening in the next reaction. One of these phosphates is getting donated to ADP to make ATP. We've made our first ATP, and not only have we made one, we've made two because we have two molecules of 1,3-bis phosphoglycerate. What I've just described to you is one of three ways that cells make ATP. There are three ways that cells make ATP. The first one is what you've just seen. It's what we call substrate-level phosphorylation. And I'm going to describe to you what that means. Substrate-level phosphorylation occurs when a high-energy molecule donates a phosphate to ADP to make ATP. The high-energy molecule here was 1,3-bis phosphoglycerate. It donated one of its phosphates to make ATP. There are two other ways that cells make ATP, and I'll briefly mention them to you, but we'll talk more about them later in the term. One is what's called oxidative phosphorylation. This is a process that occurs in the mitochondria, and I'm not going to give you a definition for that right now because it's not important at the moment. Oxidative phosphorylation. And the third method is one that you do know the name of and what it means, and it's photosynthesis. Photosynthesis does what we call photophosphorylation, meaning it uses the energy of light to make ATP. Okay. Well, we've just gone from making 1,3-bis phosphoglycerate to making 3-phosphoglycerate because we gave up one of those phosphates. And you're sitting here thinking, but Kevin, you said something about 2,3-BPG. What happened to the 2? Well, we're actually getting ready for it right here. All right? Now, if I were to tell you the name of this enzyme, you would have to know it, and I can't tell you the name of the enzyme or you would kill me, right? So I'm not going to tell you the name of the enzyme. I'm just going to tell you the name of the last half of the enzyme, and you won't even have to know that. How's that? All right? The name of the last half of this enzyme is a mutase. A mutase. What does a mutase sound like? It sounds kind of like mutant or something, right? Mutant involves change. And mutase enzymes are kind of interesting. I'm going to tell you how they work. Because if you understand how they work, you're going to understand how 2,3-BPG is made. Okay? A mutase, well, let's look at what the mutase is doing first. It's converting 3-phosphoglycerate into 2-phosphoglycerate. It's going from 3-PG down to 2-PG. Right? Well, that seems pretty simple, right? You grab the phosphate on position 3 and you move it to position 2, and bang, you're done. And that's not what a mutase does. How does a mutase work? A mutase works by first putting a phosphate onto position 2. In other words, it doesn't move the phosphate, it puts a phosphate onto position 2. At that point, what's the name of the molecule that you suppose you have? 2,3-BPG, right? And then the mutase pulls the phosphate off of position 3, and you're left behind with 2-phosphoglycerate, right? So where does the 2,3-BPG come from? Well, it turns out that that intermediate that the cell makes, where it puts the phosphate onto position 2, that intermediate is stable. And some of it will leak out of the enzyme. Some of it will leak out of the enzyme. That leakage is pretty small. There's not very much that leaks out. Most of it gets converted directly into 2-phosphoglycerate. But a small amount of it, a small percentage, maybe 1% of it, leaks out of the enzyme and comes out as 2,3-BPG. That 2,3-BPG, you know, is a signal to hemoglobin, right? And what did I say about 2,3-BPG? I said it was a signal that rapid metabolism was going on. Rapid metabolism. Where do you suppose you're going to get the most 2,3-BPG? Where you have the most glycolysis going on. And that's rapid metabolism. That's pretty cool. The inefficiency of the enzyme to convert 100% of it into 2-phosphoglycerate releases a signal that the body uses to say, hey, hold on, there's a lot of metabolism going on there. And as we will see, a lot of metabolism requires a lot of oxygen. It's a very nice, simple setup. Everybody understand that? I'm just going to stop and just stare at you guys for a minute. What's that? You think I should tell a joke? I can never resist an opportunity to tell a joke. This is a dumb joke. You want a dumb joke? Okay. Dumb joke. This is a professor joke. Okay, I can tell a professor joke, right? I won't pick on anybody. I'll pick on myself. So there's these two professors, right? They're sitting on this airplane, and they're flying a red-eye from Los Angeles to New York. Okay, and the pilot comes on and he says, "This is your pilot, United Airlines Flight 331. We'll be flying from Los Angeles." I guess it's not quite a red-eye. It ruins the joke. So anyway, we'll be flying from Los Angeles to New York. Our flight time is about 4 hours, and we will be arriving in New York at about midnight. And the two professors go, okay, so man, I'm tired. I'm going to take a nap, because I'm going to get to New York. I'm going to have to do something. So they're just kind of sitting there, and the flight takes off, and they're going a little ways, and pretty soon there's this bang! Looking around, wakes the professors up, and the one professor looks at the other and goes, what the hell was that? Pilot comes on and goes, "Oh, hello, everyone. I'm sorry to disturb you, but we've had a malfunction on engine number 1 on our plane, and it looks like everything's okay. This happens sometimes on a plane, but we'll make it to New York, but it's going to take us an extra hour to get there, so we should be arriving there by 1 a.m. Okay, go back to sleep. They're nodding off, and pretty soon there's this bang! Oh my God! Pilot goes, "Well, ladies and gentlemen, I've never seen this happen before, but our number 4 engine has just conked out, and we only have 2 engines going right now, but we can make it to New York. It's just going to delay us by another hour. We should be arriving by 2 a.m. Guys go, wow, this is really pretty crazy, And so they're nodding off, and of course, what do you suppose happens next? Well, pretty soon, bang! Pilot says, "Well, ladies and gentlemen, in the history of flying, I've never seen 3 engines actually give way, but number 2 engine is just conked out. 3 engine, whatever it was. It's just conked out. We have one engine, but don't worry, that engine will get us to New York. We'll be arriving by about 3 a.m. The one professor pokes the other in the ribs and says, God, he said, I hope that last engine makes it, because if it doesn't, we're going to be flying all night. I told you it was dumb. All right. You guys really look dead today. Somebody going to tell me a joke? Nobody ever tells me a joke. Nobody? You have a joke? Oh, maybe you could tell me later. You guys ready for happy hour? Yeah. You know what we should do? At the end of the term, we'll have a happy hour. How about that? Yeah. You want to do that? If I buy the beer? How many are 21? Most are 21. Okay. Well, we go someplace that if you're not 21, you can still get in. How's that? We'll do that. I like to do that. I do it with this class because it's small enough that I can actually go out and I'll buy the first few pictures. We'll do that. Okay. Now everybody's perked up. Yeah, let's go. It takes that to get through glycolysis sometimes, right? Well, we have to get through glycolysis, guys. And by the way, the material for the exam will likely cover through Monday. Likely cover through Monday. Okay. Well, we've just made 2-phosphoglycerate. We're almost there. We have 2 reactions to go. 2 reactions to go. The first reaction involves the loss of water. We're converting 2-phosphoglycerate into a molecule that you'll like the name of. It's phosphoenol pyruvate, for which we will abbreviate as PEP, PEP. PEP. What does PEP tell you? PEP tells you that you have a lot of energy, something that none of you seem to have right now. A lot of energy, guys. And this molecule, yes, thank you. This molecule has a lot of energy. This molecule, okay, well, you can probably guess it has a lot of energy, being the really astute people that you are, because you look here and you say, oh, there's an ATP being made. And so what does it take to make ATP by substrate level phosphorylation? It takes a high energy molecule. And so therefore, PEP is a high energy molecule. PEP is a very high energy molecule. In fact, PEP is such a high energy molecule that it releases almost enough energy to make 2 ATPs. Not just one, but almost enough energy to make 2. The cell doesn't have a way to capture that energy to make the second one. What does that mean? That energy is lost. Well, what happens when energy is lost? If you go out to your car, is your car 100% efficient? No. There's no engine that's 100% efficient. You learned that in physics, right? What's the byproduct of inefficiency? Heat, right? Heat is released. The car, you go out and you touch your engine. You don't want to touch your engine after it's been running for a while because the engine is hot. That's why you have a radiator. That's why you have a cooling system, right? Because that engine is hot, and it's hot because it's not 100% efficient in terms of converting the burning of gas to the movement of the car. So you might start asking yourself, well, does this reaction release heat if that energy is not realized in the form of making ATP? And the answer is, you're ahead of me if you think that, because that's correct. The reason you get hot when you go running or exercising is because your reactions are not 100% efficient in terms of the things that they are doing. And here's a prime reason. This guy's releasing a lot of energy, a lot of heat. This reaction, by the way, there's the last enzyme name you have to know, pyruvate kinase. This reaction is what I call the Big Bang. All right? It's not the beginning of the universe. It's the end of glycolysis, but I call it the Big Bang because it's making ATP and it's got excess energy. Big Bang. Pretty cool. Now, questions about that? Yes? The pyruvate kinase? The enzyme is called, okay, the enzyme is called pyruvate kinase. And I'm going to summarize the enzymes for you, okay? I've given you 5 enzymes. 10 reactions, I've given you 5 enzymes to know. The first enzyme was hexokinase. The reason I give you hexokinase is because we'll talk later about it being important in regulating glycolysis. The second enzyme was called PFK. And the reason you need to know that enzyme is because it's very important for regulating glycolysis. Glycolysis is an unusual pathway. We'll see that there are 3 enzymes that help to regulate the pathway. The third enzyme I said you should know was aldolase. And that was because it had that unusual reaction it catalyzed with a high positive delta G0 prime. It catalyzed a reaction that had a high positive delta G0 prime. The fourth enzyme I gave you was G3PDH. And I gave you that one because it catalyzed the only oxidation reaction. The fourth enzyme, I'm sorry, the fifth, I can't even count. The fifth enzyme I gave you is pyruvate kinase. I gave it to you for 2 reasons. 1, it catalyzes the Big Bang. And 2, as we shall see, it's important in regulating glycolysis. And here's the last enzyme of the pathway regulating the pathway. This is very unusual. We'll see why that's the case later. Are you ready for a song? I have a song that summarizes everything that you've seen. It's called The Sound of Glucose. Aldehyde sugars are always aldoses, and if there's a ketone, we call them ketoses. Some will form structures in circular rings. Saccharides do some incredible things. Onto a glucose, we add a P to it. ATP energy ought to renew it. Quick rearranging creates F6P without requiring input energy. At a high rate, add a phosphate with PFK. F16BP is made up this way, so we can run and play. Da, 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 da. Aldolase breaks it and then it releases DHAP and a few G3 pieces. These both turn into 1,3-BPG, adding electrons onto NAD. Phosphate plus ADP makes ATP, while giving cells what they need, energy, making triphosphates a situation of substrate level phosphorylation. 3-BPG, 2-BPG, lose a water. PEP gets a high energy state just to make pyruvate. So all the glucose gets broken and bent. If there's no oxygen, cells must ferment. Pyruvate, lactate, our cells hit the wall. Some lucky yeast get to make ethanol. This is the end of your glucose's song, unless you goof up and get it all wrong. Break it, don't make it to yield ATP. You'll save your cells from futility. All right, guys. See you, my